Hello everyone, in this video we shall discuss about the basic technique of animal cell culture. Animal cell culture involves the in vitro maintenance and propagation of animal cells in a controlled environment. It is the growth of cells in the lab condition where the necessary physiological conditions and nutrients are provided in vitro. It is widely used in biomedical research, drug discovery, vaccine production and regenerative medicine, and so on. The success of cell culture depends on precise techniques, sterile conditions and proper nutrient supply. Animal cell culture media requires certain preparation where initially we have to set up a laboratory. The laboratory required for animal cell culture should comprise of a laminar flow hood where it provides a sterile workspace to conduct the experiment, avoiding contamination of the microbes. Carbon dioxide incubator is one of the important equipment that have been included in the animal cell culture laboratory that maintains a controlled temperature of 37 degrees Celsius, humidity and also the carbon dioxide concentration. Inverted microscope is used for studying cell morphology, observation and even cell viability. The animal cell culture lab requires a water bath which pre-warms media and reagents to 37 degrees Celsius, which is the ideal temperature for cell proliferation and growth. Centrifuge is also used in animal cell culture cell culture laboratory, where it is used for cell collection and washing. Autoclave being the major component or major equipment in the animal cell culture laboratory, which is installed for sterilization. Sterilization is very important as it avoids the contamination of microbes during animal cell culture. Aseptic condition and following aseptic technique is very important in animal cell culture media. To prevent contamination, work in a biosafety cabinet. The biosafety cabinet is used to perform animal cell culture, and the surface of laminar airflow and the working surfaces are wiped with 70% alcohol, which steriles or which makes the working space sterile and free from contaminants. And usually the equipment and the pipettes or the gloves that we use during the animal cell culture, they are sterilized and they are used sterile. Usually when we perform the animal cell culture technique, we have to avoid talking and unnecessary movements that can contaminate the culture area. So these being the initial setup, the types of that animal cell culture techniques we have are the primary cell culture. The primary cell culture is the technique where the cells are directly isolated from tissue and they have a limited lifespan. So some of the cells that are followed or cultured through primary culture are fibroblasts, endothelial cells, etc. Secondary culture is nothing but the cells subcultured from the primary culture. After primary culture, if we subculture the primary culture, then it is called as secondary culture. Usually transformed fibroblasts and other cells are cultured through secondary culture. Continuous cell lines are immortalized cells that can divide indefinitely. So they don't have definite cell division number, they are immortal cells. Some of the examples of continuous cell lines are HeLa, CHO, Vero, etc. Suspension culture is the liquid culture where the cells grow freely in liquid medium. Some of the cells cultured through suspension culture are lymphocytes, hybridomas, etc. Adherent culture is a kind of culture where adherent cells are grown, where the cells attach to the culture dish, and epithelial fibroblast cells are usually cultured through adherent culture technique. The basic steps that are followed in animal cell culture technique is, first step is isolation of cells. Initially, the cells to be cultured are obtained from tissues, blood or any established cell lines. So when the cells are obtained or the tissue is obtained, the tissue will be undertaken for enzymatic digestion. So enzymes like trypsin, collagenase or dispase break down extracellular matrix and the cells are then filtered and centrifuged to remove debris. Mechanical disruption is also followed for tissue disintegration, where cells are isolated by mincing, grinding or scraping. So, using blood derived cells, leukocytes and lymphocytes are separated via density gradient centrifugation. So, these are the initial steps where cells are isolated from the tissue. Step 2 is cell seeding and growth, where the obtained cells are resuspended in a suitable culture medium and transferred to a sterile culture flask or petri dish. And these petri dish, after being transferred, they are placed in carbon dioxide incubator at 37 degrees Celsius for growth. And then next step comes the subculturing, where when the cells reach 70 to 80% of confluence, they are passed to the fresh medium. The old medium which is already used is removed and the cells are washed with PBS and the trypsin EDTA is added for the adherent cells and the trypsin is neutralized with fresh media, fresh complete media. Then the cells are centrifuged and resuspended in the cells in the fresh medium. So, then the cells are transferred into the new flasks for continued growth. After the growth, the cells are assessed for its quality. One of the quality assessment is through cell counting and viability assessment. Hemocytometer counting is one of the technique where the cell quality is measured. The cells which are mixed with trypan blue dye, that excludes dead cells. Dead cells accepts the stain and the live cells remain unstained, so that while counting the live cells in hemocytometer, the cell quality and the growth quality can be decided. So, automated cell counters can also be used as instruments for cell counting. After counting and viability testing, freezing and cryopreservation is carried out, where the cells are stored for future use by freezing in liquid nitrogen, like cryopreservation. In cryopreservation, cells are grown to log phase, detached, collected and resuspended in freezing medium, and after, when the cells are being used or when the situation comes where they are in use, then the cells are thawed and removed from the liquid nitrogen and they are transferred to the flask. After this, monitoring and maintenance, the cell morphology, after tested the cell morphology, healthy cells are selected, contaminated cells are removed and the antibiotics are used for less contaminated culture. So, this culture technique has importance in biopharmaceuticals, cancer research, stem cell therapy, toxicology testing, genetic engineering, etc. In these areas, cell culture technique helps a lot, where the cells cultured are used in getting several products, and the cell culture is also being carried out to study various aspects including cancer research. Thank you.